Thank you very much for the introduction and for giving me the opportunity to present what we found so far with this collaboration. Our lab is pretty new in the Trophoblast work and actually started with this collaboration with Accelerated Bioscience. So basically, before that, I want to present what the talk structure is going to be. I'm going to give you a small introduction regarding of what we're doing as a lab so far, and also a small introduction, very small introduction on NK cells. And then I'm going to present the objective of this collaboration with Accelerated Bioscience. I'm going to briefly describe the methods of what we did so far, and then the main findings regarding the human trophoblast stem cells and the induced pluripotent stem cells. And I'm going to finish with conclusions and possible future directions. Okay, so our lab is primarily an HIV lab, so we do have several collaboration with international teams, and we are basically a translational research lab focused on immunology, infectious disease, and cancer. We primarily work with innate response, and we develop also, we look to develop HIV cure-directed efforts, and also other people in the lab are working with cancer immunotherapy, and we're also doing clinical trials. As you saw in the title, we're gonna see if trophoblast cells exhibit any phenotypic or functional characteristic of NK cells. So I put a small introduction here. So what are the NK cells? NK cells are like main cells of the innate immune response, and they are defined by flow cytometry. We define them by the lack of expression of CD3, CD14, CD19, and CD20, and the expression of CD16 and CD56. So here you see a dot plot of flow cytometry when we have CD56 versus CD16. The main subset of the NK cells, the one that express both, the CD56 positive, 16 positive cells that are considered as cytotoxic. More analysis of the expression of CD56 and CD16 is shown here when they're divided as the NK cells are divided as CD56 bright and CD56 high expressing cells. So these high expressing cells, they could have CD16 or not, and they are primarily cytokine secreting cells, while the cytotoxic are the primarily CD56 bright and CD16 positive cells. And there are also described in the literature mature NKs that have CD16, but they don't express CD56, and more immature NKs that express CD56 bright but lack CD16. Besides CD56 and CD16, NK cells express a large panel of receptors, so they have activating receptors, inhibitory receptors, chemokine and cytokine receptors, and also adhesion receptors. Some of them are shown here. Binding of the receptors with their ligand triggers different responses for the NK cells. So basically, the binding of the activating and inhibiting receptors with the ligand, the target cells, results in activation of different signal pathways that can result in the activation or inhibition depending on the balance of the signal that it's transmitted after the binding of the receptors with their ligand. What is the function of the NK cells? The main function of the NK cells is the lysis of the target cells. So the way the NK cells kill the targets is dual. So they can do either by direct cell lysis or by antibody-dependent cytotoxicity. So the direct cell lysis happens when the activating receptor signal is stronger after binding with the receptor on the target cells, together with the lack of inhibitory receptor binding to the ligand, or a lack signal, not very strong signal transmitted by this binding, results in the lysis of the tumor of the target cells. In addition to that, NK cells secrete cytokines that could influence the adaptive immune system, the T cells, and those T cells can also go and kill the target cells. So this is one way of killing. The other way is by linkage of the CD16 receptor, which is one of the receptors that we saw that there is on NK cells, with the Fc receptor of an antibody, which binds to the antigen on the target cells, and that triggers the lysis of the target cells with the signal that's transmitted with this binding. So this is the antibody-dependent cytotoxicity. Our lab is primarily a HIV lab, as I said, so we did a lot of work where we presented the effects of different immunotherapy strategies on NK cells and on NK cell function. I'm going to go very quick here. I just want to show you that we are not a human trophoblast cells. We are pretty much coming from a different world. So basically, we did a lot of studies of treatment interruption. Here you see cycles of interruptions, which are shown here as ATI, and this is the period that the patients are on ART. So during all these periods initially that we studied, we saw that there was no change. Basically, there was a preservation of the NK cells, different subsets of NK cells. So here, this is the distribution of the frequency of NK cells. Here is the median, here are the quartiles. So we have different subsets that were preserved during all these cycles of interruption and restarting therapy. And also we saw preservation of the direct cell toxicity. So this led to other immunotherapy trials where we combine interruption of therapy together with immunotherapy. In these cases, we have trials where we use interferon together with interruption of therapy. And we saw that basically here, we have an activation during these cycles of immunotherapy of the NK cells, and this activation of different subsets of NK cells together with increased toxicity led to decrease of the viral load. So in this study, we saw that the patients that did not increase the viral load, that didn't have viral rebound after the immunotherapy, had higher levels of activating NK, and they also have higher levels of cytotoxicity, while patients that had lower levels that actually show a viral rebound after the immunotherapy, they had a low ability of killing, and they started with low levels of activating NK. Then we also measured in another trial the effects of combined administration of interferon with neutralizing antibodies, the bNAbs, and we saw that through these cycles we had preserved the cytotoxicity functionality of the cells during the different time points before or at the during the immunotherapy or at the end of immunotherapy, as shown here. And also we had a decrease of the antibody-dependent cytotoxicity during immunotherapy that was restored though after the end of the immunotherapy. So this was basically to show you what we are doing as a lab. So because of this work on the NK cells, we were approached by Accelerated Biosciences in order to study trophoblast cells and induce pluripotent stem cells. So for the trophoblast stem cells, we want to see if they have any phenotypic and functional cytotoxic property that is comparing to what we see in the NK cell subset. So we're asked to thaw, culture, and expand the human trophoblast stem cells, and also to do a phenotypic assessment and also measure the ability, if any, of these cells to kill target cells. We also approached to see if induced pluripotent stem cells coming again from the Accelerated Biosciences could have the potential after differentiation to support hematopoietic differentiation in immunodeficient mice secreting human cytokines, and we wanted to compare that potential, if any, with human fetal tissue-derived CD4 positive stem cells. So here we again were asked to thaw, culture, and expand IPCs, and also to culture them and differentiate them to CD34 stem cells and or NK cells, and then determine in a similar fashion as we have here the phenotypic and cytotoxic properties of these NK differentiated cells, and also see if we give those differentiated cells to immunodeficient mice that are able to secrete cytokines, if that could induce production of different hematopoietic cells. So for these purposes, we were given 3 vials of 2 different human trophoblast stem cell lines from Accelerated Biosciences, and also we're given 2 vials of 2 different IPCs clone, C1 and C6. I was responsible primarily for the first objective, and these were done by another senior staff scientist in the lab. But I'm going to present like what we have in both these objectives so far. So basically, regarding the human trophoblast stem cells, Today, I'm going to show you what we did regarding the thaw culture and expansion of HTSCs, and also what we found with the phenotypic assessment so far. Regarding the IPCs, we're going to show results on the culture and expansion, and also the differentiation of the trophoblast derived IPCs to CD34 and or NK cells. I'm going to start with the human trophoblast stem cells. So basically, we follow a protocol that were given to us by Accelerated Biosciences. We withdraw the cells and we culture them in pre-coated plates for 4 hours, and then we change the media and we left them for at least 72 hours, and we harvest the cells, and then we seed them again, or we use them for flow cytometry to assess whether they do express any NK markers. And we also use as a comparator whole blood PBMCs that were isolated from blood, and we also stain them for NK. So for the IPCs, again, we follow the protocols that were given to us by Accelerated Biosciences. And here the cells were thawed, they were put in a pre-coated plate, and again, they were harvested. And when they were at 70% confluence, they were detached either by GCDR or by acutase. The ones that were detached by GCDR, they were placed as a seed cluster to another plate, and they were seeded for expansion. While the ones that were detached by acutase, their single cells were put in 6-well plates in this concentration, 3 million per well, and they were left for 5 days to form embryonic bodies. Here you see like an embryonic body. We measure then the differentiation of these cells in coated plates that allowed for adherence of these cells, or in non-coated plates that were not, the cells were not allowed to adhere, just to see if there are any difference in this differentiation. And also we try to differentiate them to NK cells. So I'm going to start with what we found for the ITCs. So here I'm going to show you first the results from the thaw culture and the expansion, and then from the phenotypic assessment. So the main question that we had regarding the cells were if in our hands we can actually thaw, culture them, and expand them. So here I'm showing you the example of the results of one cell line, but it was exactly similar for the other cell line, which I'm not going to show. So we saw this cell line, and we saw that the viability based on the expectance of the cells was 82%, and also the viability based on all the cells that were able to count was 91%, so it was very good. And here we saw the cells at 4 hours, and this magnified version is not very strong, I know, but like this is how the picture came up. Then after 72 hours, we also saw that the cells remained viable, and they were about 80%, and they were very confluent. And then we seeded the cells, and after the first seeding in 20 hours, we noticed that we had like some floating cellular debris, and we have an overgrowth of the cells. So basically the plate was pretty much, the flask was pretty much full. And we seeded them again, and part of it we just left it in culture, and another part we froze and thawed. So the ones that we left in culture, we saw that the cells after 48 hours were almost overgrown, and they start showing some shapes that we thought that they are a little bit weird. Also, we saw cells from this second seeding, and we saw that the viability was very good and was based around 97%. For both cell lines, we saw that the viability was pretty good. So it was about 90 to 80 to 90%. So the first question, the answer was that we're able to culture and expand the cells. One characteristic that we noticed was that when we look on the culture, we had cells that were very big and some cells that were still small. And we look a little bit on the literature, we saw that other people have also reported that there are different sizes of the cytotrophoblasts. We have small or large. Human trophoblast stem cell lines also have been reported to form colonies of different size and shape. So that's what we know so far. I mean, we'll appreciate any feedback from you guys that you are more familiar with using these cells, but this is what we saw in our hands. Because our main goal was actually to use these cells in the future for cytotoxicity assays. Our experience in cytotoxicity assays, as you can see, is like from human peripheral blood cells. So these are floating cells, they're not adherent cells. So we want to see, because these cells are adherent, if they are surviving when we culture them under non-adherent conditions. So we saw one vial from both of these cell lines. And as you can see the viability was very good. It was about 85 and 80% for each one of these cell lines. And we cultured them for 4 hours under adherent conditions in a flask, but we also cultured them under non-adherent conditions. And we saw that they were not adhering, but they were still viable. Now we want to see if we leave those non-adherent cells for a longer time, if they're gonna be okay. So we culture them for 24 hours, and we saw with different densities. So here we have a 1.2 million, here we have 200,000, and here we have 400,000 cells. And we saw that after 24 hours, slowly the cells, as we increase the concentration, they start forming embryonic bodies. And 48 hours, this is even more intense, this phenomenon. We're not worried about this because our toxicity assays span for 4 hours. So then we never reach culture of the cells up to 24 hours or 48 hours, but just want to try. And we also want to see what's the effect of the number of the cells in the culture, how this affects like the trophoblast cells under non-adherent condition. So basically we saw that at 4 hours was the time that we use for the cytotoxicity assays. We can have the cells non-adherent and good viability, but if we leave them more and we have them in very high densities, they form embryonic bodies, so we cannot use them for this kind of assays. And so overall, we saw that we're able to culture and expand the cells. We saw cells with different sizes, and we also saw that we can culture the cells under non-adherent condition for a small amount of time and none in a very high density. I'm going to shift to what we saw with the phenotypic characterization of the cells. First, we want to see, because we are very new in this field, to see if they express some of these markers as we expected them to do. And here I'm showing you the results of these both cell lines, the 1905 and the 203, culture at 4 hours under non-adherent condition. The results were the same after adherent condition. I'm just showing the non-adherent condition because that's what we're planning to use in the toxicity assays if we do them in the future. So here we go. I'm showing the gating strategy. So we have the single cells, and then we go to viable cells that we use aqua live to segregate the live from the dead cells. So these are the live cells. And when we gate on the live cells and we look on the expression of EGFR, HLA-ABC, and ITGA6, we see that they do express these markers as expected. The same is for the other cell line. We had some concerns because now what we see is that if you look here, we have different cell sizes of the trophoblast cells. That was not something that we expected. We thought that everything is going to be pretty much the same size. But as I showed you before, in the actual culture, we did see large cells and small cells. So this replicates also in the flow cytometry assay. Then we want to see if they express 56 and 16, which are the main markers of NKs, together with the lack of the CD3, CD14, 19, and 20, which characterized as LIN3. So here I'm showing you the comparator of a human PBMC, how it shows when we did the staining at the same time. So we have singlet cells, we have the live cells again, and then the live cells, this is the forward side scatter, and we always gauge on lymphocytes, and then we go to LIN3, and we pick the LIN3 negative. You see we have two populations, the positive and the negative in the lymphocytes, which is expected because we have T cells and we also have NK cells. So if we focus on the LIN3 negative, we have the distribution of 56 and 16, and you have the double positive, and then you have the single positive populations as expected. So we want to see if this staining with these markers using 56 in PSI7 and CD16 before 50 is replicated on trophoblast cells. So we use again LIN3, and here again we gate on the live cells, and this is one cell line, 4 hours non-adherent condition and 72 hours under adherent conditions, okay? There are not much difference. We have more cells in the second option, but basically what we see from the live cells, we go to Lin3, everything is negative, and this is based on isotype and unstained conditions that we use, so everything seems to be negative, so we don't have any expression of CD3, CD14, 19, and 20 in the trophoblast cells, which was expected. And then we see regarding the 56 and 16, it seems that we have 56 positive, 16 negative cells. And also we have 16, a little bit of 16 positive cells. And this is shown also here. And here, forgive me, but I cannot see the bottom part of my presentation, so I cannot read exactly what the numbers here, but basically I highlighted in yellow the percentage of 56 positive, 16 negative population and 56 positive, 16 positive population in the two conditions, and also the mean fluorescence intensity. So you can see that we were able to detect these subsets, like primarily this one, in both conditions. Now, because we didn't see any Lin3 positive, we decided in the future staining not to use Lin3 and to gate directly from the live cells. So we repeated the staining. Again, here it seems that we have the different sizes of trophoblast cells. So we repeated the staining without using the Lin3, but keeping the same fluorochromes. And here we see again that we have 56 positive cells, and we don't seem to have a lot of CD16 positive cells, and we have a little bit of 56 positive, 16 negative, 16 positive cells. It is more obvious here that we have different sizes of cells, and this is because as we were learning how to use the trophoblast cells, we also change the voltages and all stuff, so we can see this population a little bit better in these stainings. And we want to explore what's going on with these three populations that we see here, if they are different in the expression of CD16 and CD56. So we gated each one of these populations. So these are the small ones, these are the transitional, as we call them, and these are the big ones. And here we see the expression of 56 and 16 in each one of these populations. So we see that the big ones seem to have more of the 56, and the small one have but less than the big ones. Okay, and here you saw the same populations in the isotype, how they behave, and also in the unstained condition. And this is showing all three. So the gray is the isotype, the red is the unstained, and the blue is the stain with the 56 and 16 antibodies. We want to see if that is something that we're also going to observe if we use a different fluorochrome for these markers. So we stain again by using now 56 V450 and CD16 APC87. And again, we gate the same way. Here, it seems that we have also a large CD16 positive population, which is negative for CD56, and we still see the 56 positive, 16 negative, and a little bit of double positive. Again, we have the three populations, so we gated them in the same way. And here we see that there is a difference, like the big ones seem to express more of the 56, and the small one seems to express more of the CD16. So again, here I'm showing you the isotype, the unstained, and all three combined. Okay, so we see that these populations, the small and the big one, seem to have a difference in the expression of CD16 and CD56 markers, with the big ones expressing mostly the 56 and the small one expressing mostly the CD16. We then move to stain cells for markers that are activating or inhibitory. There is a large panel, as you can see here. We did the staining, we started the analysis, but because in the meantime we were finding these three populations of NKs, we kind of stopped the gating because we wanted your feedback if we should proceed with exploring this a little bit more or not. So I'm not going to show you the results for this staining, although we did perform that staining. And conclude that basically we're able to detect trophoblastic stem cells markers and 56 and 16 in the trophoblast stem cell lines that were given to us. We did observe different sizes, and this caused one problem in the collection in the flow cytometry because it clogged the machine sometimes. And also it started a question for us because we saw cells that they have different sizes, and with the smaller cells having more CD16, which means they have more CD16 FC receptor and maybe more potent in an ADCC assay, while the larger cells are having more of 56 marker. So here we culture the cells, and here I'm showing you that the culture was okay and was confluent as expected for the IPCs single cell seeding or the cluster that I show you in the methods. And we're able also to solve them and differentiate them to CD34 stem cells. So here you see the undifferentiated, and here you see the ones that do express CD34 with a lack of CD45 or CD43. So we're able to differentiate them to CD34 stem cells, but not to NK cells yet. So we had some issues on that. So basically, we were able to solve and expand the IPCs, and we are still working on the NK cell differentiation. So basically, we're able so far to culture and expand the cells and characterize them, and we show the different sizes in the trophoblast stem cells. So the hypothesis that the stem cells, the trophoblast stem cells that have smaller sizes are more potent maybe in ADC activity than the ones that are bigger in size. And we are targeting completion of the phenotypic and functional characterization of both cell lines, the trophoblast and the IPC. So I would like to thank Chinsun and Matt, who did the staining and culture of the cells, and also Jess. And Robert Jejuan was responsible for the IPC work. And Heather started this collaboration with Accelerated Biosciences. Of course, Luis, who is in charge of the lab and the center and mentor all these years. And everybody from Accelerated Bioscience. I hope I didn't forget anybody.